We were also in the other room where we saw like the centrifuge, the PCR amplification. I guess all these machines. Do you think you can give us a quick overview of how that equipment comes into play?
Yeah. So the equipment's an important part. So the things we do, I mean, the techniques that we use a lot are gene expression profiling.
Right.
Which, so in a cell you have DNA that codes all the information, and then some DNA is transcribed into RNA.
Right.
And those are the active genes, and then that RNA is turned into protein. So we think the RNA is sort of a nice level of information you could obtain about the state of a cell. So a popular technique that Stanford's one of the earliest places to develop in the mid-1990s is gene expression profiling on microarrays. We have a microarray chip that has basically all the genes in the genome, spots for all of them, and you can quantify levels of expression of each gene in the genome. So we've done a lot of gene expression profiling of different sarcoma types in this lab. So that's one technique that's driven a lot of the research. Other ones include immunohistochemistry. You know what that is?
I can only guess from the name.
So that is, there's a lot of archival tissue, tumor samples that are embedded in wax and fixed in formalin. And immunohistochemistry is a technique that you can use on this type of archival tissue. And what it does is it allows you to measure the expression of different proteins. So gene expression profiling is RNA, and this is protein. So the findings from the microarray experiments can inform which proteins you're interested in looking into using immunohistochemistry on tissue microarrays. So whereas a gene microarray, you look at one sample and you look at like 40,000 genes the expression of. Okay. Tissue microarray is actually a glass slide where you have samples from maybe a few hundred different patients, and you look at different proteins one at a time. So the first technique, you're looking at sort of one patient, all the genes, and the second technique, you're looking at lots of patients, but only a few proteins.
The PCR?
Oh, so what is PCR?
Yeah, maybe.
Or like the role it plays
so PCR in molecular diagnostics plays like an essential role. This is, I sort of work in this research lab, but I also work as a molecular pathology fellow in a molecular diagnostics lab.
Okay.
And basically every test we do starts out with PCR. So it's absolutely ubiquitous in the molecular diagnostic lab. And the way it works is basically all it's doing is amplifying a region of DNA of interest. And you can do reverse transcription PCR where you turn RNA into DNA and then amplify that type of RNA. So basically you're just making lots and lots of copies of whatever gene you're interested in. And then usually there's a second, some kind of downstream assay that you do with all those copies of whatever gene you're interested in, like sequencing or different probes.